Hello, everyone. So I'm doing a voiceover of slides live. So when we go over this, there may be some pauses. Feel free to fast forward through the pauses. So today we're going to look at signal transduction pathways as the screen loads and how they maintain something called homeostasis. And homeostasis, please write this down, is when organisms work to maintain things at a constant level. Okay, and please make sure you write that down. And we're going to look at an example of this, and we'll be looking at other examples, but we're going to look at an example with regulating blood sugar. Okay, so again, homeostasis is the ability to change something back to a normal position if it goes too high or if it goes too low. So we're going to look, and there's going to be sounds. So it's, for example, if a body condition goes too high, there's a gland which will release a hormone, and then that hormone will lower the body condition. And I'm just going to take the sound off. I actually don't want the sound, so give me a second. No sound now. Now, the important thing to realize here as we click through the sounds again is that the hormone is a ligand that responds, that lowers the body condition through a signal transduction pathway. So if you didn't get that, please rewind and rewrite that the hormone signals the body to lower the condition by using a signal transduction pathway, and a hormone is an example of a ligand. Now, the body condition can also go through too low, and the same thing will happen, but it will be another gland that releases a hormone that raises the body condition through a signal transduction pathway. Now, and this is called a negative feedback model. So whenever there is something that you're returning back to a normal level, if something goes too high or too low, that is called a negative feedback model. So we're going to be focusing in this video on the regulation of blood sugar. Okay, so your blood has, when you eat something that is sweet, has sugar in your blood. If it goes too high, the cells get stimulated to store glucose as the polysaccharide glycogen in the liver. So if your blood sugar goes too high, and you might be thinking about diabetes here, if blood sugar goes too high, the body will be signaled, we'll see how that signal works, to take up the sugar and store it in the liver as glycogen. Now, similarly, if the blood sugar goes too low, the body will be signaled. So again, there's also a reverse possibility that this could go in the opposite direction. If the blood sugar goes too low, then you can release the glycogen stored in the liver. Please make sure you wrote at least two things on this slide. So again, this is the feedback loop, and I'm just going to get the pen out. This is the feedback loop for blood sugar. So blood sugar should be about, if you measure your blood sugar, if you have a meter, should be about 90 milligrams per 100 milliliters of blood. If that level goes too high, the pancreas releases a hormone known as insulin, and then insulin will return the blood sugar back to normal by stimulating the body cells to take up sugar. It also causes the sugar to be stored in the liver as glycogen, and it reduces your appetite. So that's what happens if your blood sugar goes too high. The important part is that it stimulates your cells to take up glucose and store it as glycogen or use it in the body cells. Now, similarly, if the blood sugar goes too low, the pancreas will release a different hormone known as glucagon. And glucagon does the reverse. It causes the cells to release the glucose in the liver that's stored as glycogen. So instead of storing it as glycogen, it's going to stimulate the liver to release glucose. It also triggers hunger. So please make sure that you wrote at least two things on this slide. Okay, so now we're going to be looking at a signal transduction pathway that stimulates cells to take in glucose. This is a large slide, and whenever I give you a large slide, I want you to write all over it. So this here, insulin is binding to a receptor. So this here is reception. Remember, it triggers, I can't write reception, but you guys can write reception here. I don't have my pen with me, unfortunately. So I can't write, but this is reception. It causes a conformational change in the receptor, which activates this protein, which is called the PI3 kinase. Do you need to remember it? No. Okay, but they may describe the pathway and they may name the P13 kinase. Remember, it's a kinase, so this kinase is going to activate the ATK kinase. Again, okay, that doesn't mean that you need to memorize this, but you need to be able to read pathways like this. Then the ATK kinase activates the GLUT4 vesicle protein to insert itself in the membrane. Now, glucose, you remember, is a charged particle, so it can't enter the cell unless the GLUT4 protein is there. So without insulin being bound to the receptor, this entire pathway won't happen. And if this entire pathway won't happen, glucose can't get into the cell and the blood sugar will remain high. Now, to label the parts of the signal transduction pathway, this is reception. This here is transduction. And this here, when we insert the GLUT4 protein into the membrane, this is response. Okay, so this is what happens when blood sugar is too high. Insulin will trigger the cells to take in glucose through this pathway. Now, what happens if glucose goes too low in the blood? Well, then the hormone glucagon, this here is glucagon, is released. That activates the G protein. So this here is the G protein. The G protein then activates the enzyme adenylyl cyclase. Adenylyl cyclase is an enzyme, and it turns ATP into a molecule known as cyclic AMP. Cyclic AMP, you need to write this down, is a second messenger. It amplifies the signal or makes the signal stronger. That you must write down. So if you didn't write it down, make sure you write it down. Cyclic AMP activates protein kinase A, which is abbreviated PKA. That activates phosphorylase kinase by adding phosphate groups, which activates phosphorylase by adding phosphate groups. Phosphorylase is the response. Phosphorylase breaks down glycogen into glucose, and then glucose can enter the cells, can go into the blood and enter the cells. Okay, now that was a lot. I suggest replaying it, okay, and writing down what happens at each step. I am going to repeat it a little bit, making sure to label the parts of a signal transduction pathway. So this here, the glycogen bonding to the receptor, this is reception. This whole thing with adenylyl cyclase and the kinases is transduction. Okay, remember, second messenger cyclic AMP serve to amplify the signal. These are kinases. This is a phosphorylation cascade. These are kinases that activate each other by adding phosphate groups. And last but not least, the response is that this final enzyme that is activated can turn glycogen into glucose, and then that glucose can be released into cells. So again, here we saw how if blood sugar gets too high, the cell signal transduction pathways can signal it to be turned into glycogen and be stored in the liver. Similarly, if blood sugar gets too low, glycogen can be released, broken down into glucose, and released into the blood to make the blood sugar go back In class, I believe on Friday or Monday, depending upon how quickly we get things done on Friday, we are going to discuss how this relates to diabetes, and we're going to do some practice multiple choice in class. So thank you for listening to the video. Hopefully this video works. I'm going to discard my annotations, and I will see you in class on Friday. And as I end the recording, I am going to make sure that hopefully this worked, and I will see you guys on Friday.